let's move on with another aspect of the foundation of biochemistry. Now we are moving to the genetic foundation of biochemistry. The genetic foundation of biochemistry. Now, earlier on, while we were discussing the biochemically relevant characteristics of living thing, I said living thing has the ability to replicate or to give birth or to give rise to a faithful copy of itself. So we're going to look at that to a little extent. We are going to expatiate a little bit on that, although that's another topic on its own in biochemistry, where you study molecular genetics, the study of heredity, your DNA metabolism, RNA metabolism, protein synthesis. That's a wide topic in biochemistry. But for our introductory class, we still say one or two things about it. So let's get started. Living organisms reproduce a faithful copy of itself as a result of conserved molecular structure of genetic information. This is what I'm trying to say. Inside every cell, inside every living and viable cell, there is a molecular structure that helps the cell to conserve genetical information. Are we together? And this genetical information is passed from generation to generation. So what's the molecule that help in conserving this genetical information? They are known as nucleic acids. Nucleic acids. Nucleic acids. And your nucleic acids are, they can either be DNA, which is your deoxyribonucleic acid, or it can be your RNA, ribonucleic acid. Although it is your DNA that will get transcribed to RNA. The difference between DNA and RNA is not far-fetched. Just two things. In your DNA, for you to have a nucleic acid, maybe I should take my time to explain that. Nucleic acid, okay, let me take time to explain this. Do you remember I told you that cell has the ability to form supramolecular structure? All right, having remembered that, now look at this. Your cell has the ability to create, or pardon me, your cell has the ability to match sugar, your cell has the ability to match sugar with nitrogenous base and with phosphate group. To give you molecule known as nucleotide. Now, this sugar plus nitrogenous base without phosphate group is what we know as nucleoside. We are going somewhere. Sugar plus nitrogenous base gives you your nucleoside, but nucleoside, which is sugar plus nitrogenous base plus phosphate group, gives you your nucleotide. Now, nucleotide will now merge. Your cell has a way of condensing nucleotides together to give you what? Nucleic acids. Now, nucleic acids now occur in strands. They are complementary strand, double helix strand. It coils together to give you your double helix strand, which then supercoils to give you your chromosome. Are we together? Now, this nucleic acid, which is the polymer of nucleotide, is what constitutes the molecular structure that helps in retaining inheritance information, or that help in retaining genetic information that is passed from one generation to the other. Now I said nucleic acid can either be DNA or RNA. Let me explain it. If it is DNA, the sugar you are going to have here will be deoxyribose. And if it is RNA, the sugar you will have will be what? Ribose. We go to the in-depth explanation here by the time we are explaining nucleotide, by the time we are explaining the genetical aspect of biochemistry. Now, let's leave it there. Now, if you are having a DNA, your nitrogenous base can either be deoxy, okay, can either be deoxyadenylate. Okay, let me not go. I don't want to bore you with too many biochemistry facts so that it will not look confusing. Let me just do it this way. Your nitrogenous group in DNA will either be adenine, guanine, thymine, and cytosine. Yeah. Now these two are known as purine, and these two are known as pyrimidine. Let's leave it this way. The rest of the explanation on this is a little bit complex for an introductory class. Now, for ribonucleic acid, your nitrogenous base will be adenine, guanine, cytosine, and pardon me, thymine. And uracil. Because cytosine, the difference is that in your RNA, cytosine will not be seen. What you'll be seeing is uracil, okay, because of that. So this forms the structure of what this DNA and RNA is, the molecular structure that helps in conserving genetical information from one progeny to the descendant, then down the generation lane. So another thing I must mention is that genetic continuity is embedded in each DNA molecule. Now, in your cell, you have a nucleus. In the nucleus, you see a chromosome. See a chromosome? Yeah, usually drawn like this. Now, by the time you unwind your chromosome, you see chromatin. Now, chromatin, by the time you unwind it, you notice that your chromatin is made up of nucleic acid supercoiled together with protein known as histone protein. Histone. And there are 5 types of histone protein. I won't go into details of that now. Let me go through what I've said again. In your cell, you have the nucleus. In the nucleus, you have the chromosome. All right? Your chromosome is thoroughly supercoiled and overwound over each other. Now, if you try to unwind the chromosome, you get your chromatin. Now, you notice that your chromatin is a polymer of nucleic acid, which is complexed with proteins known as histone protein. Now, by the time you now stretch out your the DNA, the nucleic acid, pardon me, by the time you stretch out the nucleic acid in your DNA, you just notice that it is a sequence of nucleotides polymerized together, and the chain is very long. All right. Now, in the length of your DNA, you now have genes. Within the length of deoxyribonucleic acid, you have genes. So what are genes? Genes what are genes? Genes are segments of DNA that code for a particular character. Genes are segments of DNA that code for a particular character. In biochemistry, we have another name for calling genes. They are called exons. Now let me explain this better. In your DNA, let's say I have A, C, okay? Our DNA will always start with a particular code, AUG, C, G, U. Pardon me, you see U alone in RNA. T, G, A, C, A, G, C, C. Now, look at this. Assuming this is a nucleotide, and only this nucleic acid, oligonucleotide here is a short segment of nucleic acid anyway, oligonucleotide. Now, here, assuming this is what I find in my chromatin. Now, it is not everything here that will be coding for a particular character or for a particular protein. Now, I may have, because it occurs in codons, in triads. Now, this, okay, only this, in this chain, are we together? In this chain, it may be only this that is coding for a particular character. Now, this segment that is coding for a particular character in my nucleic acid is my gene, otherwise known as what? Exon. So, the coding part, the coding segments, the segments of a nucleic acid that code for a particular protein or for a particular character is what I know as my gene. The non-coding parts are known as introns. The fact that they are non-coding do not mean that they are useless. These non-coding parts, the parts that they are not coding, does not mean they are useless. More often than not in biochemistry, emphasis are laid on introns. Initially in the knowledge of biochemistry, emphasis used to be laid on exons. But in modern biochemistry, it has been found that emphasis, more often than not, needed to be placed on introns because on most occasions, they regulate exon exhibition. They regulate exon expression. That's a little bit advanced in biochemistry, so I'll stop at that level. So note that in cell, heredity is conserved. How? Nucleic acid, which is the chemical structure or the chemical apparatus that help to relay information from one generation to the other, is conserved with a high degree of fidelity in the cell. So in our cell, there are ways, there are mechanisms that help in conserving the integrity of nucleic acid. We usually call it the halves of nucleic acid, so that nucleic acid, which helps in transferring genetic information from one generation to the other, so that the integrity of nucleic acid will be conserved in the cell. What will the cell do? There is a way of replication. That's our first R. Replication. So through replication, the cell, through replication, the nucleic acids in a cell, in the nucleus of a cell, has a way of reproducing itself, a faithful copy of itself, not just reproducing itself at that. It has a way of reproducing a perfect copy of itself. How? in the cell, the nucleic and the DNA specifically. Let me concentrate on DNA. We'll talk more about this when we are talking about the central dogma of molecular biology. Now let me come back to this. How is replication possible in a cell? In the nucleus of a cell, the DNA occurs in twin like this, in a double helix form. Alright, so the information on one strand, let me call this strand A, let me call this strand B. Now the information on strand A is complementary to the information on strand B. That's where Chagaf's rule comes to play. Chagaf tells us that A will base pair, because between one strand and the other there's what we call base pairing. A will base pair with C, and G will base pair with C. Now the double link here is showing us hydrogen bond. There are two hydrogen bonds between A and C. I told you what A, C, G and C meant. They are the nitrogenous base: adenine, thymine, guanine, adenine, thymine, guanine, and cytosine. Okay, so adenine, the nitrogenous base in your DNA, in the first strand, the adenine nitrogenous base will base pair with thymine, the guanine will base pair with cytosine. Don't worry, we take this completely, we take it explicitly by the time we are doing DNA metabolism, nucleic acid metabolism. Alright, so here, the information on strand A is complementary to the information on strand B, such that peradventure, if in the course of transmission of genetic information, or in the course of mitosis, if the information on one strand is lost, on one strand is lost because of the information on the complementary strand. For instance, the information on my strand B here is lost because I still have intact information on strand A. I can still reproduce back this information on strand B by a process known as repair. There's a repair mechanism that will help me to bring back the information here. But how will it know what to bring back? Based on the information on the adjacent complementary strand. So I'll go through what I've said again. Genetical information, which we have said is nucleic acid, conserved nucleic acid in the nucleus of an organism, is the molecule that helps transfer information from one generation to the other. How does it do it? Replication. This nucleic acid has the ability to unwind and produce a faithful copy of itself. So starting from this place, if there is an unwinding, this can create another copy. Let me use another marker so that it will be clear. At this place of unwinding, there can be a replication. This can start another copy. So in the 5' to 3' direction, I can create, the cell can create another copy of its complementary strand. So at the way this occurs, so at the end of the day, I'm going to have two of this base paired to this, and another one of this base paired to this. So this is what I'm trying to say in essence. This nucleic acid here, from the open end, replication can occur such that from the open end in the 5' to 3' direction, replication can occur such that the cell will produce a copy of a complementary strand of each strand in your previous DNA. So at the end of the day, you are going to have two DNA molecules, one old strand and a new complementary strand. The complementary strand will now be the new or the nascent strand. But in the course of this replication, information may be lost, but the information that is lost does not mean all hope is lost. There is a mechanism for replacing back lost information. How? Based on the information you are having on the complementary strand, the one that is lost can be recovered. So these two mechanism, repair and replication and repair, help in the continuity and the fidelity of the nucleic acid composition of a cell, which means when the nucleic acid composition of the cell is well preserved, the genetic information of the cell is well preserved. So another thing I will go a little bit into the molecular central dogma of molecular biology now, that the linear sequence of DNA encodes protein in their correct folding. Now, I gave an example here earlier on. Now look up. Now I said here I have gene. A gene is the segment of DNA that's code for a particular character. Now, how will this code for character? What I have here, for instance, is my DNA. Now, for my DNA to code for a character or to code for a protein, it has to go through this. It has to go through this sequence. My DNA will get transcribed to RNA. Here I have transcription. And my RNA can get translated to protein. So, the linear sequence here in your DNA will help to encode protein. How? The DNA is first transcribed. The double-stranded DNA is first transcribed by a process known as transcription to RNA, which is single-stranded RNA. I'll go through that again. Double-stranded DNA is transcribed to single-stranded RNA. And for this purpose, we are talking about the mRNA, messenger RNA, because we're having three major types of RNA. We have the mRNA, which is the messenger RNA. We have the rRNA, the ribosomal RNA, and we have the tRNA. Now, the RNA is then translated to what? Protein, which is translated to protein, or the physical phenotypical character that you see an organism exhibit. Okay. So this is very important. It's very important. Now, DNA can produce another copy of DNA by the process of replication. By replication, DNA can produce new DNA. But DNA, when DNA is to be exhibited as a character, or to be exhibited or to code for a protein, the DNA will first of all get transcribed to mRNA, and mRNA is translated to protein. All right. For now, let's take a hold on that and let's move to the evolutionary foundation of biochemistry.